Hi, today we're going to be talking about the circulatory response to exercise, something called hemodynamics, how you control your blood flow, and ultimately we'll talk about blood pressure and the control of exercise blood flow. So to do this, first off, I want to review how blood flow will influence your aerobic capacity or your VO2 max. And then we'll talk about all the different things that influence blood flow during exercise. And one of the things that will become very important and a concept that you need to understand is this one over here, which relates to how you distribute your cardiac output during exercise. Your heart can only pump so much blood. And if we dilated everything, if everything in your body got all the blood it wanted, it would far outpace what the heart can do. Your heart can pump probably about 25, 30 liters of blood per minute. But your arteries and capillaries throughout your body, if your arterioles dilated maximally, they would demand like 200 liters per minute, right? So we have to be very deliberate about where we send the blood at rest and during exercise. So we've talked in the past about the Fick principle, where where your VO2 can be calculated as the product of cardiac output, noted as a Q with a dot above it, and the arterial venous oxygen difference. And there should be an overbar here to represent that the venous sample is an average of all the veins, a mixed venous sample. So your VO2 is strongly influenced by these things. So your cardiac output, how much blood your heart pumps every minute, is very strongly related to your VO2 max. The higher your cardiac output, the odds are that you'll have a higher VO2 max. And that's what's illustrated in this diagram right here. Extremely tight relationship. Sorry about that. And in this, on the arterial venous oxygen difference, we've talked about what that represents in the past. The arterial venous oxygen difference represents the difference between your CaO2 minus your CvO2. And that's just how much blood you have, or how much oxygen you find in about 100 milliliters of blood. And so the more oxygen that we deliver to the muscle, the better. And the more oxygen that the muscle takes out of the blood, the better. And so we'll talk about how these things are influenced by blood flow. So your blood flow can be described by something called Poiseuille's law. And in this law, you might have learned this in physics, flow through a tube is influenced by the perfusion pressure. That's what this triangle P, delta P means. So the perfusion pressure would be how much pressure is at the start of the tube versus how much pressure is at the end of the tube. And in our case, the pressure at the start, this would usually be generated most of it by your heart. So a lot of it would be like your systolic and diastolic blood pressure. And the pressure at the end of the tube, this would probably be your venous pressure, pushing back, your back pressure, or sometimes your intramuscular pressure can create that pressure gradient there. But the higher the difference, the greater the difference between the pressure at one end to the other, the faster the flow. So if you want to increase your blood flow, your heart rate can increase, cardiac output can increase to drive up your blood pressure, and that should help push blood through the arteries a little bit faster. Something that's even more influential is the radius of the tube that you're perfusing. So right here, the bigger the tube, the faster it goes. And what's impressive about this is that small changes in your radius will have really big influences on your blood flow. Because this is raised to the fourth power, that means that a little change will cause a big change in flow. Just to put that in perspective, if we doubled the perfusion pressure, so if we times it by 2, which we never really do, the blood pressure doesn't go up that much. But if we did, that would result in a doubling in blood flow. If we double the radius, so if we dilate this, make this tube twice as big, that would increase your blood flow by 16 times. it is so much easier to increase your blood flow by dilating than it is by increasing the perfusion pressure. And what's more is that in order to increase the perfusion pressure, as I mentioned, a lot of that relies on the heart. So if you're trying to drive up your blood flow just by increasing perfusion pressure, you're going to put a lot of burden on the heart, which costs a lot of energy in ATP. Whereas dilating will not cost you a lot of energy, and it actually makes the job of the heart pretty easy. So dilation in most circumstances is the preferred method of increasing blood flow. Now, the other things in this equation, the N here would be the viscosity of the blood. You could imagine if you're perfusing, trying to put blood through a tube versus trying to put jello or cement through a tube, it's going to be how much pressure you require to get through the liquid through the tube is going to differ. So the more viscous the fluid, the slower the flow. And this can change in some circumstances like dehydration, and it might change a little bit with exercise, but not too much. So most of the time we just ignore it. The length of the tube, also, this is not going to change that much in terms of your vasculature during exercise. But if you have a really long tube, you'll have to push a lot harder to get the flow through. So flow will be slower. So another way to look at this isn't on this, but let me clear this up. Another way to look at your blood flow is that flow, let's see if I can do this. Flow is equal to the perfusion pressure divided by what we call resistance. And things that increase resistance will be viscosity. If you have a high viscosity or a high length, it's going to cause a lot of resistance to flow. Things that decrease resistance are the radius. So really our resistance is just the inverse of this. So it'd be viscosity length over radius to the fourth. This is what resistance is. And when you exercise, you're going to dilate, so you're going to decrease resistance, and your blood is going to go through the path of least resistance. So if you have really low resistance, that means you need very little pressure to get the high blood flow. But if you have really high resistance, let's say you don't dilate very well, or you're pumping really viscous blood, that's going to mean really high resistance to flow, which will mean in order to get the same flow, so the same amount of blood and oxygen delivered to the muscle to support a VO2, in order to get that, if you have high resistance, you have to have a really high perfusion pressure, which is really going to come down a lot to the heart. So blood is going to go through the path of least resistance, and what determines the resistance is the radius. That's the most influential thing. Other things will influence it, but the most influential thing is the radius of the vasculature. And it's not just the size of your aorta or the size of your like your femoral or your brachial artery. Actually, the resistance that's most pertinent here is actually the radius that's most pertinent is the size of your very, very tiny arteries, the arterioles within your muscles. They're tiny, but when you add them all up together, it's a very big radius, especially if you can double them. They have a high capacity for increasing their size. So what does vasodilation look like? Your smooth muscle in your arteries, we'll start here in a neutral scenario. Your smooth muscle in your arteries overlaps almost like in a diamond pattern here. And so that would result in a lot of, like, just you got a bunch of smooth muscle here. That's not getting the point, but you got a bunch of smooth muscle over there and they're kind of thick. If you dilate, you relax the smooth muscle. So now it elongates. Your actin and myosin don't interact as much, so your muscle goes to a longer position. And that results in the artery expanding outward. Now, if you want to vasoconstrict, you're going to increase the overlap between your muscle cells and your actin and myosin and all of that. And so you get a thicker, bulging smooth muscle with a very small internal lumen. So the relaxation or the contraction of your smooth muscle is what's going to cause the artery to get bigger or smaller. And And the arteries that have the greatest capacity for changing their size are what we call your arterioles. Now, your aorta here is a really big one. It's about, I don't know, about an inch or so big, depending on your body size. And then you have these conduit arteries, like your brachial artery or femoral artery. They're also pretty big. So if you think about Poiseuille's law, where the bigger the radius, the greater the flow, you're like, wow, the aorta must be like where you control your blood flow because it's the biggest artery in your body. But there's only one aorta. As you keep going down this vascular tree, what happens is that you see more and more vasculature. Let me double check and make sure. Okay, I'm screen share recording. I'm good. I was worried that I wasn't recording. So as you go down from the aorta to conduit arteries, you split. So the aorta is clearly the biggest. Then you got these conduit arteries. They're a little smaller. Then you got these things here that we would call feed arteries. These are the arteries that go off of like the bigger conduit arteries and then dive into the muscle. They're a little bit bigger and they can change their size some. And then here, these tiny things, these are what we would call the arterioles. They're really small. And then the very last one, the purple, we would call the capillaries. And they actually have pretty much no capacity to change their size. The flow through them is just completely dependent on what's pushing into them upstream. So I do a lot of research in this area, and on these conduit arteries, they can dilate a little bit. I've seen them dilate maybe 10% during exercise, 20% if you're lucky in certain scenarios, but that's it. I did some research on these feed arteries, where we got some from people when they were going through surgeries. Usually surgeons take out these arteries as they're trying to do whatever the heck they're taking out, and they would throw them away. Instead of throwing them in the garbage, I asked the surgeons if I could have them, and they gave them to me willingly, and the people agreed. And we tested how much these could change their size, and they can double, right? So this is like maybe 15 to 20% change in size on those conduit arteries. But these feed arteries can go up 100% is what we saw. And then these arterioles, these are really small and really hard to get from people. But in animals, it's been shown that these things can at least go 100% to maybe 200% or more change in the size. So much greater capacity to change size. And if they're all really small under resting conditions, they're going to cause a really high resistance to flow. But if these arteries here, your feed arteries and mostly your arterioles, if they dilate, the resistance to flow is going to plummet, very low resistance, which means the heart over here, as it's pushing blood, it's just going to fly through the aorta and the conduit arteries really quickly because there's little to no resistance downstream. Now your capillaries, these capillaries here, they're a single layer of cells like the endothelium, and there's no smooth muscle on those things. They have no capacity to really change their size. So they're pretty much fixed. Your control of blood flow is happening here. Mostly in the arterioles, to a little limited extent in the feed arteries, but mostly in the arterioles. And the trick here is that the radius that matters is not just the radius of one little artery, but the radius of the whole vascular network in your area of interest. So if you want to know like what's controlling blood flow in your leg, it's not the size of your femoral artery usually it's dictating your blood flow in your leg. It's the size of all of your arterioles together in your leg that is going to dictate the flow. The bigger the radius of all your little arterioles, the lower the resistance and the greater the flow. So that's the radius that matters, not just of one single artery, but of that whole level in the vascular network. So what determines blood flow during exercise? Here's a fun diagram. It's a little small, I apologize. But what this illustrates is flow through your aorta at rest. So that would really be your cardiac output. So at rest, your cardiac output somewhere around 5 liters per minute. And when I do research and measure blood flow through the femoral artery at rest, it's somewhere around half a liter per minute, probably a little less, right? So your heart has no trouble perfusing your leg at rest. The cardiac output is far greater than what the legs are wanting. When you do maximum exercise, if you're like a very fit, elite, like highly endurance trained person, you might see cardiac output go up to 25, 35 liters per minute. This is really high levels. Most of us are probably around 20, 25 liters per minute. And under those scenarios, we would see about 10 liters per minute through the femoral artery. You got 2 of those femoral arteries, so about 20 liters per minute of your cardiac output just going through your legs. So here it seems like, okay, the heart's able to keep up. The heart's probably dictating how much blood is going through the body. But now look, even during maximal exercise, all your resistance arteries, all of these are not maximally dilated. They're still constricted to a certain extent. If we dilated everything, all the resistance arteries in your body, this is what it would look like. Your maximum cardiac output is still the same, somewhere around 35 liters per minute. But if we dilated all your resistance arteries, in order to keep them perfused, you would need somewhere around 250, 225 liters per minute. Now, this is an estimate, but you get the idea that your heart cannot keep up with how much the arteries could demand. So you have to budget them. If the arteries, all of them got all the blood flow they wanted, this is how much the heart would have to pump. And it can't, right? So the heart's stuck down here. Because of that, you have to restrict the arterioles and the vasculature to not getting everything they want all at once. You have to budget where the blood flow is going. So what you'll do is, let's say that this vascular bed is like your quads and you're doing some cycling exercise. So you might dilate that area. But if you're going to dilate that, you have to constrict these to reduce blood flow to that area to make available blood for your quads. If you dilated everything, you get in this scenario where you're outstripping the heart, your blood pressure would plummet, you'd pass out, and it wouldn't be good. So your body is very deliberate, at least a healthy body is very deliberate on what it dilates and what it constricts. Even during maximum exercise, your quads, I don't think they'll be fully dilated. You'll still have a little bit of restriction. We've done studies on this and found that there's still some vasoconstriction in these arterioles during maximum exercise. The body is holding them back so that they can't deliver as much blood to them, because if they did, then all the blood would go there and none would go to your brain and the rest of your body, and you'd be in really bad shape. So they restrict them a little bit. So now that brings up the point, how would you budget your allocation of cardiac output? Let's say that you got 35 liters per minute. What things are you going to prioritize at rest versus what things will you prioritize during maximum exercise? So at rest, you'll get a lot of your blood flow going to your gut for digestion, the kidneys for filtering blood and all of that. We decrease how much your blood flow goes to the kidneys and the gut during exercise, and increase how much goes to the muscle. You also want to make sure you prioritize always blood flow to the brain and blood flow to your heart. So you don't want that to decrease, because they're active during exercise as well. So you're going to have a lot of blood flow distributed to the muscle. About 80% of your cardiac output is going to go to the active muscle during exercise, intense exercise, or during like an endurance exercise. And then the remainder of that, a big chunk of it will be going to your heart, and a big chunk of it will be going to your brain. Your body prioritizes keeping blood flow to those two organs as well, for obvious reasons. But you're going to decrease blood flow to other areas like your digestive system and your kidneys. And then if you exercise in the heat, that places an additional burden on you. You have to divert blood to your skin to dissipate the heat. And that means if you're diverting blood to your skin, that's blood that's not going to your muscle. And so you get less blood flow to your muscle, which can decrease your exercise capacity in the heat, just because you're diverting blood away from the muscle to go to the skin. So this diagram illustrates just a rough idea of what happens during exercise. So your muscle blood flow is going to go up a lot. So this shows like 1,600% increase in muscle blood flow, while blood flow to your splanchnic region, which is really just like this, the gut and the kidneys kind of idea, it's going to go down about 20% or so, maybe more, depending on the organ during exercise. All right, so how do you determine vasodilation during exercise? What causes these arteries to vasodilate or vasoconstrict? We talked about the smooth muscle, that it increases the overlap or decreases the overlap of actin and myosin, and then the overlap of the smooth muscle cells. But what dictates that? Now let's put this in perspective. I already talked about this a little bit, but when you exercise, your muscle blood flow can go up 15 to 30 times, right? It's a huge increase in exercise. But when you exercise, your blood pressure definitely does not go up 15 to 30 times. It probably doesn't even go up double in most of you when you're doing endurance exercise. So the majority of our increase in blood flow is happening because of dilation of the resistance vasculature. And very little of our change in blood flow is going to come because of changes in viscosity or length. You'll get a little bit due to viscosity. We can talk more about it on another day if you want, but yeah, for the purpose of this class, let's just assume most of the time that it just doesn't change unless otherwise stated. So how is the size of your arterioles regulated? So here's a really simplified diagram of what your arterioles might look like. You got this part here would be the smooth muscle. So that's where we have the overlap of the smooth muscle cells with your actin and myosin. And then this outer layer is called your adventitia. It's kind of like an encasement. And then the very inside here is the endothelium. This is a single layer of cells on the very inside of the vasculature. And for years and years and years, it was thought that it was just a lining. But as I'll show you in a minute, it's actually much more than just a lining. It's very crucial for regulating your blood flow. So your smooth muscle in here with your overlap between the smooth muscle cells and your actin and myosin, it has receptors for norepinephrine on there. And if your sympathetic nerves, they go onto your arteries, your arterioles especially, and when they're releasing their norepinephrine, it will bind onto receptors on the smooth muscle and cause contraction. So whenever you have a sympathetic response, most of the time, in most vasculature, you're going to have norepinephrine being released, which will cause contraction. And so if you get contraction because of the norepinephrine, you're going to get vasoconstriction and the size of the artery will decrease. When you decrease the size of the artery, that's going to drive up blood pressure. So when you get stressed and have this fight or flight response, or you're really embarrassed or nervous for a test, one of the things, or let's say you have white coat syndrome, when you're getting blood pressure done at the doctor's office and you get really nervous about that, you're having a lot of norepinephrine being released and it's causing vasoconstriction. So the radius is getting smaller and you're driving up your blood pressure a lot higher. So that's sympathetic control of your diameter or your radius, which in most of your vascular networks, the sympathetics are going to cause vasoconstriction. So in the arterioles in your muscle and in most of your organs, sympathetics will cause vasoconstriction. The exceptions to that would be in your skin. You can have sympathetic vasodilation in the skin, which we'll talk about when we talk about thermoregulation. And you can also have sympathetic vasodilation in the coronary arteries, so the arteries of the heart. Pretty much everywhere else, the sympathetics is going to cause vasoconstriction. Now, the smooth muscle, or sorry, the endothelium, this single layer of cells, as I mentioned, for years and years and years, everyone thought it was just a lining, that it didn't do much. But now it's recognized that it's extremely active in the control of blood flow and in the health of your vasculature. In fact, a lot of my research focuses on this endothelium. And what's understood now is that as that endothelium goes bad, as you age, it goes bad. There's just a single layer of cells. It's very vulnerable to changes, to damage. And if it goes bad, your control of blood flow goes bad. And also, your risk for cardiovascular disease increases exponentially as this little layer of cells goes dysfunctional. Under normal circumstances, that endothelium is responsible for preventing clotting, preventing atherosclerosis, and lots of other things, and also for dilation and constriction. And if it starts to go bad, now atherosclerosis goes unabated. It just goes without being resisted by anything. So the endothelium is extremely important. And so what happens is this endothelium release vasodilators. One of the most common and well understood right now is nitric oxide. And it will diffuse from the endothelium to the smooth muscle and cause relaxation. So norepinephrine causes contraction. Nitric oxide will cause relaxation. And if you relax the smooth muscle, the artery expands out and you get dilation. So here's what your vasculature looks like in like a muscle. So this is from some kind of animal. I can't remember which one, but this would be an arteriole. And then you got these long capillaries that run along your skeletal muscles, interfacing with them. And the more interface you get between the capillary and the exercising muscle, the more chance you have to diffuse oxygen and other things like glucose. So you want to have a lot of capillaries overlapping with skeletal muscle. But the capillaries do not control blood flow at all. They're just passive participants as we understand it right now. And all your blood flow is controlled, most of it upstream, by these bigger arterioles. Now in your textbook you might read, or in other textbooks you might read, that you have something called precapillary sphincters, which they're just like this little smooth muscle that goes around the capillary that turns on and off capillaries so they're not perfused at rest or exercise, and then they open up during exercise. This was hypothesized by a guy named August Krogh, who's an absolute genius. Based on the information that he had and the capabilities that he had like 100-something years ago, it was an excellent hypothesis. But it went untested and accepted as basically as gospel for years and years and years still. But now as technology is caught up, there's really no evidence for these precapillary sphincters within the muscle. They are not there. The control of blood flow happens up here in the arterioles. These precapillary sphincters are non-existent. So when you get to med school or other schools and your professors are talking about the precapillary sphincters, just know they're really not there. Or they might talk about how you don't have blood flow going through most of your capillaries at rest. You do. Most of your capillaries at rest have blood flow. So these ideas of precapillary sphincters and not perfusing parts of your muscle during rest, it's just not there. It's not a high rate of perfusion. So here's another a YouTube video showing you how drastically blood flow can change just by dilating. Now remember, your blood flow is dictated by the perfusion pressure and vascular resistance. So in this preparation, what they're doing, they have probably like a hamster muscle or something. They're looking at it under a microscope, and they start off by vasoconstricting everything. So they expose all the vasculature to something like norepinephrine that causes it to vasoconstrict. And then they give it a dilator, adenosine. So this would be basically like a product of metabolism. As you break down like your ATP and other things, you'll see adenosine levels increase. So they expose the muscle to adenosine, and you'll see what happens to the blood flow. So under these circumstances where they vasoconstricted and you can see that there's very little flow. There's still red blood cells flowing through all the capillaries. So this would be like a larger arteriole, the big thing that you see. But now as we apply adenosine, look at all these red blood cells moving in the background. You can see a lot more going on. Now, this is not a physiological condition. You would always have at least a little bit of flow going through all those capillaries. But now that we're dilating here in that, sorry, I can't point at it. Now that you're dilating those arterioles upstream, you get red blood cells just flying through the capillaries. And so blood flow goes up a lot. So by decreasing the resistance in the resistance arteries, so in your arterioles, you're going to drive up blood flow throughout the whole muscle. And I love this video because you can just see the individual red blood cells flying through their capillaries. You can see that some of them actually have to like squeeze through the capillary. It's really cool. So how would you preferentially distribute the limited amount of blood flow to active muscle? So you remember when we talked about exercise and glucose distribution, that you only have a little bit of glucose in your blood. And insulin is a hormone that causes glucose uptake pretty much at all muscle and everywhere. And so you reduce insulin secretion during exercise and only take up or primarily take up glucose via contraction-induced or exercise-induced glucose uptake. So you reduce the glucose uptake everywhere throughout the body and only selectively increase it in the area that needs it. That's kind of what's going to happen with your blood flow as well. You only have a limited amount of blood flow, and you have to be very deliberate about where you send it. And so the first thing that happens during exercise is you get this sympathetic signal. You are going to have sympathetic nerves that innervate pretty much all of your arterioles. Those sympathetic nerves will release norepinephrine, and the norepinephrine will make its way to the smooth muscle and cause them to vasoconstrict. That way you're decreasing blood flow to everything, pretty much everything, the exception being your heart and your skin with the sympathetics. But this sounds like a terrible idea at first because if you're exercising your legs, you need blood flow to go up to your legs. But here you have this sympathetic signal causing the release of norepinephrine that's going to try and drive down blood flow to your legs. So that sympathetic signal to decrease blood flow is even happening in the active muscle, but it is opposed by something called sympatholysis. So the sympatho, meaning sympathetic, and then the lysis, it means like a cutting or a blunting or a blocking. Probably a better description than cutting or blocking is really an overriding or outweighing. So what's going to happen in this scenario is when you're in your active muscle, you're going to get that sympathetic signal. You're going to have norepinephrine released, and it's going to be going to the smooth muscle. But simultaneously, the exercise itself is going to produce metabolites that we've talked about all semester. They're going to produce heat, hydrogen ions or acid, ADP, tension, friction. All these things stimulate that small endothelial layer, your endothelium, and it senses that, all those metabolites and other byproducts of exercise. And the exercise is going to stimulate the endothelium, and the endothelium will release dilators. Here, EDD means endothelium-dependent dilators. And the most prominent, or those are at least the most well understood right now, is nitric oxide. And in the face of the norepinephrine signal for contraction, you have these dilators signaling for relaxation. And it ends up being a balance where you have norepinephrine signaling for contraction, and on the other side, you have these EDDs, or endothelium-dependent dilators, like nitric oxide, signaling for dilation. They're both present. And in a healthy person, the signal for dilation with nitric oxide and all your other endothelium-dependent dilators should win out and heavily outweigh the signal for contraction. But in dysfunctional endotheliums, like with aging or smoking or lots of other diseases, your ability to release these dilators is blunted. So during exercise in unhealthy people, you can actually see this signal being much smaller than normal. And then the norepinephrine signal would win out and you would get more contraction than you would. And so these people would have reduced blood flow. If they have a dysfunctional endothelium, they'll get reduced blood flow to the exercising muscle, much and less dilation. So that means blood pressure is going to go way up. So they'll have an exaggerated blood pressure response to exercise as well. So this balance between the norepinephrine and the endothelium-dependent dilators, a lot of it comes down to calcium. So your smooth muscle contraction, similar in many ways to your skeletal muscle contraction, comes down to calcium. Calcium is a signal for contraction in both of those. And norepinephrine is going to result in more calcium in the smooth muscle, promoting contraction, whereas a lot of these endothelium-dependent dilators is actually going to result in less calcium within the smooth muscle cell. So really, calcium is at the center of this, of the tug of war, the balance. One signaling for increased calcium, one signaling for decreased calcium, and whichever signal is stronger, that's going to influence what ends up happening. And however much stronger the signal is, that's going to influence how much calcium is in the cell. So this idea of sympatholysis can be tested and shown experimentally. So this is a study that one of my friends did a long time ago now. They measured femoral artery blood flow, that's what this is, during exercise, so like a legs kicking exercise. And they measured it in two conditions. One where they did a control where they just infused saline. So this would be like the control or the placebo. And in another, they infused a norepinephrine mimetic, something called phenylephrine. It's basically just a sympathetic mimetic. And so you can see at rest, if you infuse the norepinephrine mimetic, blood flow goes down quite a bit. And a low intensity exercise, you infuse that, blood flow still goes down. But once you get to these higher intensities of exercise, the impact of that norepinephrine is gone. It's blunted, it's stopped. The norepinephrine was still infused in this scenario, but the signal for vasodilation due to the exercise was so great that it outweighed the signal for constriction, and it had really little to no effect. This wasn't even that intense of an exercise. This was only like 40% of their maximum capacity. So exercise in healthy individuals will cause the endothelium-dependent dilators to outweigh your signal for constriction. And in that way, you get blood flow increasing in the exercising muscle. But in your not exercising muscle, you don't have the metabolites and other things produced. So in your non-exercising muscle, you're going to have the norepinephrine go unopposed. So here, this is a non-exercising muscle. The norepinephrine is a much bigger signal than the nitric oxide and other dilators. So it's going to outweigh it, and you'll get constriction or reduced blood flow in the inactive tissue. But in your active tissue, well, that's weird. The norepinephrine signal is still about the same, but your dilator signal is much greater, much bigger. And so it's going to outweigh it. Roughly the same norepinephrine signal is going to go all throughout your body. And what's going to influence if you dilate or constrict in that area is how much of it's offset by your nitric oxide and endothelium-dependent dilator signal. Now, I keep saying nitric oxide. It's definitely not the only dilator. There's several other things like prostacyclin or prostaglandins that come into play to help with exercise dilation. In fact, we're not entirely sure of all the dilators, how blood flow goes up during exercise. That's some of the stuff that I've studied. And no matter all the things that we block with different drugs, we have a really hard time preventing the entire increase in exercise blood flow. So that's just a long way of saying we don't know all the things that contribute to exercise blood flow or the dilation. But nitric oxide appears to be one of them, and it's just the most well studied. So what are the endothelium? Oh, here we go. What are the endothelium-dependent dilators? So nitric oxide, I've said many, many times. It's extremely well described. Some people got the Nobel Prize for describing it about 20 or 30 years ago. So the nitric oxide, in addition to dilating arteries, it also is really important for other things like inhibiting atherosclerosis and thrombosis. So it's going to, the more nitric oxide you have around, the more you're going to inhibit the development of plaque in your arteries and the development of clots. Exercise training, healthy diet, these things promote a healthy endothelium, which will have a good basal level of nitric oxide around. So if you have a good lifestyle, there's a good chance you have a lot of nitric oxide floating around that's going to try and slow down the development of atherosclerosis and thrombosis. Additionally, the more nitric oxide you have around, like you're going to increase the production during exercise and other scenarios, and that also acts as a signal for angiogenesis, which is the development of new capillaries and other vasculature. So nitric oxide is extremely important, not just to dilation, but also the overall health of your vasculature. Other factors like prostaglandins and hyperpolarizing factor, these are other ways for dilation. And if you're interested in these, you can come talk to me and I'll totally geek out about it. There's some really cool stuff. Now, in my research, some of the stuff we do is we study blood flow, and we know that nitric oxide causes dilation of the arteries, and these things also cause dilation, and they also play a role in this, like inhibiting atherosclerosis and other things to an extent. So we use the artery's ability to dilate. We can measure change in blood flow very easily. So we use the ability of the artery to vasodilate as a window into how healthy the artery is. And what I and several other people much better and smarter than me who've been doing this a lot longer, what has been found is that your ability to dilate your arteries during exercise and other things is strongly predictive of your risk of cardiovascular disease and cardiovascular death. And if we can improve the ability of your arteries to dilate, simultaneously, we're improving these things, which reduce your cardiovascular risk. So that's where a lot of my research focuses, on how we can improve artery health, the ability to dilate and control blood flow, and decrease risk of cardiovascular disease. Now, finally, the arterioles, that determines how much blood goes into the capillary, how quickly it goes through the capillary. But you have to get the oxygen out of the capillary again. And this is where Fick's law of diffusion comes once again. So we talked about Fick's law of diffusion, getting the oxygen through the alveoli into the capillary. And that same concept is going to apply getting the oxygen out of the capillary into the muscle. So you have to have this pressure gradient. And the higher the pressure of oxygen in the capillary, the faster it's going to diffuse. And the lower the pressure of oxygen in the muscle, the faster it's going to diffuse. So you want to maximize that pressure gradient, really steep pressure gradient. And if you have like poor lungs where you're not getting very much oxygen into the blood, that pressure gradient is going to be smaller because you have less pressure of oxygen in the blood. So you want to maximize that pressure gradient to get a high rate of diffusion. And then you also want to maximize the area for gas exchange. So that is going to be how much the interface between the capillaries and the muscle. And as illustrated in this, the capillaries just don't like go and touch the muscle once and then go away. A lot of times they're just winding around it, just wrapping the capillary the capillaries wrapping the skeletal muscle, really trying to maximize the amount of area for gas exchange. And it goes even further. It's not just the interface between the capillary and the skeletal muscle, but it's the main influence is like how densely the capillary is filled with red blood cells. And the more red blood cells you can send into that capillary, that's going to maximize your area for gas exchange even more. So that's what's going to influence your diffusion. And the area for gas exchange, highly influential. And then you have also this idea of the membrane thickness, or how far the oxygen has to diffuse from the capillary into the muscle. And if you have like swelling or edema, if you have really low capillary density, these things might impair your ability to diffuse or get the oxygen out of the capillary into the muscle. In healthy people, it seems like this membrane thickness doesn't impact it too much, but there definitely are scenarios in pathological scenarios with disease where that membrane thickness can be a hindrance. So most of the time, what we're really trying to do is maximize the area for gas exchange. So if endurance training, you're going to see angiogenesis or an increased number of capillaries. That's going to increase your area for gas exchange and increase your ability to diffuse the blood flow faster. So endurance training also is going to increase, your rate of blood flow, how quickly the red blood cells are moving through the capillary. And if you don't increase your diffusion simultaneously, you have the chance that these red blood cells make it all the way through the capillary without getting rid of all their oxygen, because they're just moving so fast, the blood flows really fast. So you have to also maximize how many capillaries you have to slow down the blood flow, so you can distribute the red blood cells throughout more capillaries and slow down the flow. And also just reduce whatever resistance you have to diffusion. Decrease your membrane thickness. You want to maximize your perfusion pressure. All of these things need to happen in order to get the oxygen out. If you just increase your blood flow without changing your ability to diffuse, you're going to see that you're not going to get as much oxygen out as you normally would. And so your VO2 max won't go up as much as you might expect. Because you might not be able to get all the oxygen off of the red blood cells, because the red blood cells are leaving the muscle before much faster now. So simultaneously with your increase in cardiac output and simultaneously with your increase in blood flow, you also have all these other things that are increasing your ability to diffuse the oxygen out very quickly. So all these things take place to help contribute to your VO2 max. All right, that's it for today.